our last talk is going to be by Kristine Alford also from Oregon State. We've got a big contingent from OSU this time, so that's really awesome and much appreciated, all our northern partners. So talking a little bit more about the dam removal process and dissolved oxygen. So that's for you.
Thank you. Thank you. All right, hi everyone. I am a second-year PhD student at Oregon State University, also advised by Desiree Tellis and co-advised by James Peterson. So I'm going to be talking about short-term changes in dissolved oxygen downstream of the dam removals, specifically the Iron Gate Dam. So leading up to the dam removal, there were some concerns raised about the potential for dissolved oxygen to draw down to levels that could be considered fatal for some fish. And some of the questions that we were asking, and I could hear echoed from others, were, would dissolved oxygen drop down to low levels that could be dangerous for wildlife? How long would these drops last? And how far downstream would they occur? And we did not have the resources to answer all of these questions, but we did have the resources to explore some of them. But before we get into that, let's talk about some of the drivers associated with the drawdown process that can lead to declines in DO. So there are a couple of abiotic drivers associated with the reservoir drawdown process. One of these includes hypolimnetic water, or that anoxic layer of water common at the bottoms of lakes and reservoirs. So when those channel openings at the bottom of the dams occurred, there was potential for this anoxic layer of hypolimnetic water to travel downstream without being aerated first. Additionally, there's another abiotic mechanism to consider, and this is COD, or chemical oxygen demand. There were some reduced sediments present in that reservoir bed sediment, so like unoxidized iron. So there was potential for these minerals to become suspended during drawdown and then bind to oxygen as they move downstream, effectively taking it out of the water column. Additionally, there is also biological oxygen demand to consider. So just like those reduced minerals, there were also high concentrations of organic matter present in the reservoir bed sediment. And so when these particles become suspended, they create this like all-you-can-eat buffet for a host of microbes that will show up and start feasting on it. Just like us and just like fish, these microbes often breathe oxygen to survive. So the more organic matter is present, the more these microbes are eating, the more oxygen can be taken out of the system. So what my team set out to do was identify any drops in DO associated with reservoir drawdown, and then parse out whether these mechanisms were driven by biotic processes or abiotic processes. So I want to add a disclaimer here that we just finished data collection a couple of weeks ago. So I'm just going to show you some super preliminary stuff. But what my entire study did access were 3 sites. We have our furthest upstream site is 2 kilometers below the Iron Gate Dam. We also sampled 30 kilometers and 40 kilometers below the Iron Gate Dam. This farthest downstream site is right above the Shasta River confluent. At all of our sites, we were able to access continuous measurements. A lot of these were courtesy of the Kootenai tribal efforts and accessed by RES. And so these were dissolved oxygen, temperature, and turbidity at our Iron Gate Dam and our Shasta site. And we were also able to access discharge at Iron Gate. Additionally, at our middle site here, 30 kilometers below the dam, we deployed our own mini dot so we could get DO and temperature measurements. We also took weekly field trips, which what consumed that budget Will was talking about. But we did drive down about 15 times from the initiation of drawdown to collect weekly data, that is dissolved oxygen, temperature, and turbidity using a YSI handheld sonde. We also collected samples to bring back to the lab for analysis every week to assess suspended sediment concentrations, particulate organic matter, dissolved organic matter, and we also conducted 5-day biological oxygen demands of water samples every week. These samples had bed sediment, and we also had samples without bed sediment, so we could separate the sediment oxygen demand from the suspended sediment and the biological oxygen demand. So with all that said, we just got all of these data back 2 weeks ago. So what we're going to look at today are continuous dissolved oxygen measurements just at our Iron Gate Dam and our Shasta site. And we'll be looking at our weekly particulate organic matter, BOD5, and weekly DO data just at our Iron Gate site. All right. So these are those, again, provisional data. Don't tell Mike he's still here. These are these provisional data. And we see date here on the x-axis. This is from the onset of drawdown. On the y-axis, we have continued DO measurement at Iron Gate in blue and Shasta in pink. And what we were able to find as we looked to identify those drops in DO are 4 instances at which dissolved oxygen appears to drop relative to background levels. Now, as an extra little side quest here, I did want to make sure that these drops were not driven by changes in temperature. So to kind of rule out temperature as a driver of these drops in DO, I did use a Garcia-Benson model, which only takes temperature, salinity, and atmospheric pressure as inputs, and then models predicted DO. And so we see here at Iron Gate our observed DO in dark blue and our predicted DO based on those model outputs in light blue. There is a gap, so obviously there are more inputs to oxygen rather than temperature and atmospheric pressure. But so we do see a gap in these data lines here, but we don't see any of those DO drops modeled in those predicted data, which allows us to parse out or separate temperature as a driver. And I also wanted to make sure that that gap was not because of drawdown, but instead of because of just the inability of that model to correctly estimate oxygen. So I did apply it to the months leading up to drawdown too. So all that to say, those 4 drops are not driven by temperature. So based on the preliminary data that we have so far, we're going to look at 2 indicators to help us parse out whether these oxygen sags were due to biological or abiotic mechanisms. And the first indicator that we're going to use is just the patterns of drop and recovery. So typically when a DO sag is driven by abiotic processes, we see those sags occur and recover very quickly and linearly. Whereas DO sags associated with biological oxygen demand typically have a slower onset, and they occur and recover more slowly and less linearly. So just taking that and applying it back to this DO plot that we're looking at, we see 2 instances here, the first and second circles, occur pretty quickly. They appear to be almost instantaneous if you just look at the graph, but they do occur very quickly and linearly. Whereas our second 2 drops occur and recover more slowly and less linearly. I also want to point out that the second circle, that second drop that occurs on January 27th, there is about a 4-day period between the onset of the drop and full recovery, whereas that third drop that occurs on February 2nd, there's about a 12-day period between the onset of the drop and full recovery. So let's look at biological oxygen demand. We've got another plot here that represents date on the x-axis. Observed DO is on the y-axis at Iron Gate in that dark blue line. Each vertical line here represents a biological oxygen demand incubation that we conducted. Now, these were all conducted at 8 degrees Celsius to better mimic those kind of average late winter, early spring temps in the Klamath. And each dark purple dot represents the initial DO in each incubation. The light purple dots represent final DO in each incubation, and the numbers typed out at the bottom are the concentrations of DO consumed in each incubation, and those are in milligrams per liter. So a couple of things to point out here. One, for about the first half of our study, from the onset of that first drop until we get to that 4th drop here, almost all of the oxygen in our incubations is consumed. Whereas after we conclude that 4th drop in DO, for about the second half of our study, only about half of the oxygen in each incubation is consumed. But another thing I want to point out here is that these data are not super representative because oftentimes we were sampling under low DO conditions. So for example, here we were in the river on January 28th. Our BOD5 is 1.56. That's because the initial DO in our sample was 1.56. So it was all consumed, but our BOD5 does look really low. Again, these are very provisional. I'm going to be later extracting hourly DO because we did conduct these incubations using mini dots. So we have access to continuous data in those incubations. This is just what we have for today. But in order to kind of better visualize these data for myself, I did take the percentages, which I added in the third column on this table of the dissolved oxygen consumed in each incubation. I also added a column to represent particulate organic matter that we were able to assess via lab analysis. And what was interesting here is that our 4 highest occurrences of particulate organic matter all occurred during an instance of a drop in dissolved oxygen. And yeah, so I'm going to show you this, and we're going to go back to this table here. We've got our first 2 sampling trips have really low concentrations of organic matter. And during these times, all of our sampling sites were dominated by large sediments, so gravel, cobble, boulder. Then when we get up to over 3,100 milligrams per liter of organic matter, this is what we see. And somebody was asking about sediment downstream. This happened or persisted for maybe 4 weeks after the Copco adit tunnel was blasted, and it has started to move out. So at our lowest downstream site, we were not able to find any sediment present. That was right above the Shasta River confluence for about the last 3 or 4 weeks of our study. This site right here, at the top corner, that site collected more sediment than our other 2 sites. That sediment persisted pretty much throughout the entirety of our study, but the sediment level did decrease slowly over time. We did also see a layer of sediment that persisted right below Iron Gate Dam, just present in the eddy. But again, just decreasing over time. So we were able to see recovery. So we also have particulate organic matter. It's plotted here on the y-axis in the stark green line against weekly DO and the light blue line. So these weekly DO data are not those continuous data we were looking at before. These were taken with our YSI handheld sonde at the time that we grabbed our POM samples. So we can see what appears to be a pretty strong inverse correlation between particulate organic matter and dissolved oxygen. So as we're looking at how POM influences BOD, we can't rule out the effect of temperature on BOD. So we did conduct our last 3 incubations at 5 degrees Celsius, 15, and 25. Now, those are not represented on this BOD graph that we were looking at earlier. They're separate from that. And what we're able to see is a pretty strong relationship between temperature and BOD, which makes sense because microbial rates typically increase with increasing temperature. But what's interesting to see is we have really high BOD5s around like between 10 and 11 when there are low carbon present or low organic matter values present. Because at all of these temperature controlled incubations that we have on this bar graph, we're at low organic matter levels, like less than like 30 milligrams per liter here. So under those low organic matter conditions, but high temperatures, we have BOD5s at around 10 to 11 milligrams per liter, whereas under high organic matter but low temperatures, like here, when we're incubating at 8, but our carbon values are quite high, we have about 10 to 11 milligrams per liter. So there is opportunity here to explore how temperature and organic matter might interact to drive BOD, which I'm excited to kind of dive into this summer. So with all that being said, based on these preliminary data, my current hypotheses are as follows. It looks like we have about 8 days after that January 11th Iron Gate Dam fully opened. It looks like we have maybe a pulse of that hypolimnetic water coming out and keeping oxygen at a level that was slightly lower than those background levels. Then on January 23rd, we saw that Copco one adit tunnel blasted, which was really exciting. But it looks like we did see another dissolved oxygen sag just a few days after that. It looks like that could have been maybe COD associated with that high sediment concentration, maybe hypolimnetic water or a mixture of both, but it does look like this is consistent with typical abiotically driven oxygen sags. It does recover, and about a day after it recovers, it slowly drops down again, this time persisting lower levels before recovering 12 days later. This is more consistent with what you would typically see with a biologically driven oxygen sag. And since we did start to see sediment deposition there with those high concentrations of carbon, it's very possible that that was BOD. We also see another sediment pulse here on February 23rd that also contained another spike in carbon. So that's also potentially another BOD-driven dissolved oxygen sat. So again, these are just my hypotheses based on these preliminary data, and they're gonna inform the way that I kind of process the rest of the data that we collected over the summer. So I have a lot of next steps, no pun intended. I need to finish compiling all the data at all of my sites. I am going to grab our dissolved organic matter and our sediment oxygen demand data, and I'm gonna further dig into those BOD data so that I can get hourly BOD rather than 5-day. But just like Will, I'm super open to feedback here. If you have any feedback or suggestions, or you're looking at this a different way, many of you have been here and doing this a lot longer than me. So I'll be here for a little bit after the conference if you guys want to chat, and I'll also open myself up to questions. But first, I want to thank my awesome advisor, Desiree, for helping make this possible, my other advisor, Jim, and our great team.
Questions from the room? I'll start with one. They were talking a little bit about the final cofferdam removal in September-ish or sometime this fall. There'll be higher water temperatures then. And so I'm interested in if you have kind of thoughts about what might happen to dissolved oxygen under those conditions. And then a follow-up is, are you planning to study that?
Yeah, I don't have any plans to study that right now. That's one of the reasons we started to add those high temperature BODs towards the end of our study. I chose 25 very intentionally because that looks like kind of the peak of summer temperatures in those reaches in the Klamath River. It looks like while there is still sediment deposited at the sites, that carbon level is much lower. So it's possible a lot of that sediment that was there made its way downstream, or that carbon just got packed down, as it does. So even if temperatures rise, we might see a spike in BOD, but I think we might see something occur more if there's a storm that resuspends that carbon that might be packed down in the sediment. But that's just my own hypothesis.
Great presentation, thanks. Just
Yeah, thank you. We looked at some profiles, some temperature profiles recently, but I didn't look at any DO profiles. But yeah, that's a great point. We did see that drop in dissolved oxygen that didn't seem to be associated with an influx in sediment. So I think there's an opportunity to kind of explore that. So thank you.
So you're talking about kind of pulling apart the chemical. How else do you do that?
Yeah, great question. If I had unlimited money, I would have done a COD too. That was like the big glamorous part of this would have been to conduct a COD analysis, but that just really wasn't in the cards for this study. Yeah, so I think what I'm going to start doing now is just really pulling out that BOD data, getting more frequency in those BOD data, and also
hoping to calculate some decay rates. So I won't be able to tell you definitively whether these drops are caused by BOD, but I will be able to hopefully provide more detailed and more frequent data that can help us maybe be more informed about the mechanisms that are driving those drops.
This one seems to have faded.
Can't hear questions from you.
Yes, we apologize. The second microphone was dying. So I'm sorry about that for the Zoom folks. All right. Last call for questions. Kristine, thank you. That was super interesting to hear. So, all right.